it's co-funded and through governance structures is actually a college entity. And the value of that for us is firstly, it embeds the clinical trial activity within education and training of specialists throughout Australia and New Zealand. But also, of course, it very nicely aligns with opportunities for whether it's guideline development or clinical practices around our regions and beyond. The ANZCA CTN began as the Master Trials Group back in the mid-1990s. The very first NHMRC grant we got was for the Master Trial, and we published that trial in Lancet 2002. We've gone on to be really, I think, nicely successful through NHMRC and developed bigger and I think better randomised controlled trials. They're labelled under anaesthesia, but they're much more around perioperative medicine. In other words, patients undergoing often high-risk surgery who are exposed to all sorts of complications in both the hours, days, and months and even years after their surgery. The first two of these trials, in fact, have had over 1,000 citations each. And all up, these trials have enrolled well over 20,000 patients, 20,000 patients for patients having major surgery. And I'll talk about some of those trials in just a minute. We're also currently conducting 4 current NHMRC funded trials, also enrolling close to 20,000 patients, addressing various areas of practice in the partly anaesthesia but much more perioperative setting. Now, the Be Aware trial was a trial that we designed and conducted to look at awareness under anaesthesia. And it is a very rare situation. It occurs in high-risk settings, less than 1 in 100 patients across major surgery. At the time, it was 1 in 1,000 patients, but it's devastating. So patients wake up during the operation and they're paralysed and they can feel and hear everything. It's an incredibly disastrous and distressing circumstance, so much so that some patients end up with lifelong severe PTSD and have an aversion to ongoing medical care. And I've had patients who've had previous awareness who've refused cancer surgery because they literally would not or could not go through that again. Now, anaesthesia's got an amazing history of itself. It's gone on we've had it for over 160 years. It's often considered the most important advance in modern medicine. It's enabled, obviously, near pain-free labour for women in the labour wards, but perhaps even more extensively, all types of surgery for all types of conditions have happened. The thing about it, and this is our dirty little secret, is that anaesthetists have never known how to measure how deep the anaesthetic is. So we have this great knowledge of pharmacology and applied physiology. We're very good at modifying what we believe the anaesthetic depth is, but literally it's just based on best guess population information. And there was no reliable, validated equipment or monitor that could do that. I was aware of that issue many, many years ago. We identified it in a large patient satisfaction study that it was one of the most feared complications of people having surgery. I was told that if I did any research in awareness or an anaesthesia monitor, that it would ruin my research career, so I took on the challenge. We actually did the trial. It was really an interesting trial. We enrolled over 2,500 patients. 200 of these patients had actually had awareness previously, and they consented to be randomised to a monitor or standard care. That alone I found really, really interesting. The data from our trial, which was published in the Lancet, was in fact used by the company, the manufacturer of the device, and achieved FDA licensing, where it had marketing approval as the first validated monitor for awareness in anaesthesia. And I can tell you, across all of intensive care medicine and all of anaesthesia, despite the millions of clever devices we use, this is the only randomised trial that's ever validated any monitor in surgical practice. Despite that, when I sent it to the Lancet, they initially rejected it on first round review. So I wrote this appeal letter and it worked. So there you go. In 2012, in the UK, the NICE recommendations came out with a clear statement that the biospectral index depth of anaesthesia monitoring is recommended as an option in patients who are deemed at risk of awareness. That was based entirely and only on our trial, really, despite obviously using other sources of evidence. The Australian New Zealand College of Anaesthetists, and in fact the American Society of Anaesthesiologists guidelines now recommended access and use of these machines again in so-called high-risk settings. And of course there's been a Cochrane review. Now we did a survey of practice before starting this trial, and we also included in that what the beliefs of anaesthetists were about such monitors because of all the failures of devices in the past, and also their uncertainties. And we published that before starting this trial. And at the time in Australia in high-risk surgery, this monitoring was used in at most 2% of cases. We know from our recent data that in fact for major surgery right across Australia and probably New Zealand and beyond that region, it's now used, and it's certainly at least 50% of all surgeries. So we know it has had a major impact on practice. One of our other large trials is the ACTA trial. It was a factorial trial for patients having coronary artery surgery, and we're looking at two separate interventions. Simple everyday drugs used in cardiological and cardiac surgical practice. Aspirin, of course, is an antiplatelet drug, which clearly reduces thrombotic events in the out-of-hospital setting, but of course comes at a risk of bleeding. And in cardiac surgery, that has been a great concern. Tranexamic acid is an antifibrinolytic drug that actually reduces bleeding complications, but there was a concern that it increased thrombotic events. So patients with coronary artery surgery at risk of myocardial infarction and stroke and death, of course, we had these quite different drugs, different mechanisms, ideal setting for factorial design. We'd done, again, a survey of practice and showed there was marked variation in practice both through Australia and around the world, such that around 50% of centres would stop aspirin before surgery and others would continue, and 50% were using antifibrinolytics at that time. The trial was difficult for various reasons, including, as some of the discussion mentioned earlier, changes in practice that were otherwise occurring, but nevertheless I think we nailed a good result in the end. The other good thing about this was a nice, it was a difficult trial. We had to get cardiologists involved, cardiac surgeons involved, to some extent blood banks, haematologists and others, and of course cardiac anaesthetists. And even in our own hospital here at the Alfred Hospital, where we had 6 cardiac surgeons, there was at least 10 opinions about whether or not these drugs should be stopped or started in their particular patient, because they had this great gut feeling that they knew the answer all along. So we had all that difficulty, and yet I think we did a really great result. So across 31 hospitals, 7 countries, and I think overall quite nicely. And just for the tranexamic acid results, which are the more recent results, where in fact there was great concern about it would increase the risk of thrombotic events, even though it might reduce blood transfusion, we clearly demonstrated the safety of this drug. And so much so, in fact, even looking at separately myocardial infarction, there seemed to be a reduction in myocardial infarction. And there's good reasons for that. The bottom line being, if you have a higher need for transfusions, including platelet transfusion during surgery, you have a prothrombotic state. And in coronary artery surgery, that's actually a bad thing. So we're avoiding transfusions in part to reduce that. And that might be the mechanism of how it worked here. More importantly, we showed that in fact the blood sparing effects were much stronger than anyone had thought. There have been lots of little studies that had shown reduced bleeding or blood loss, but never been definitively shown to actually reduce blood transfusion itself. And more importantly, the need for reoperation after major surgery, which is a big deal of itself. If that happens, people have a 10-fold increase in mortality in cardiac surgery. So this is a fairly cheap drug. We now argue that a higher dose than has previously been used should be used, and in fact there's effectively almost no contraindications to its use across all of cardiac surgery. So that's our final recommendations for both of those. In fact, there are great international guidelines for cardiac surgery. It's the commonest major surgery in the world. It's highly expensive, it's very costly, and complications occur frequently. The normal transfusion requirement is 30 to 40%. So it's high resource major surgery. So there's lots of data, lots of guidelines, and we're anticipating changes or updating or upgrading of these guidelines on the basis of our ATTICUS trial, but that's yet to come out. I'll now just fondly speak about the RELIEF trial. This is also a fluid trial. This was only published less than 2 months ago in the New England Journal of Medicine. This was simply patients having all types of major abdominal surgery, where in fact there's been a big push over the last 5 to 10 years to use restrictive IV fluids. Use a lot less IV fluid because too much IV fluid meant tissue edema, poor wound healing, poor recovery, and a systematic review, including a Cochrane review, said that was a shorter hospital length of stay and less complications with restrictive fluids. Now, I simply didn't believe that result. It was based on a number of small trials. I thought they were quite biased. They pulled very heterogeneous endpoints and came up with recommendations that I simply didn't believe. So thankfully, we were funded. In fact, we were the top-ranked project grant in that year for NHMRC. The trial occurred on time, on budget, 3,000 patients across 47 hospitals, 7 countries, including in the US, which was rather nice. And we were effectively working against all these international guidelines that are being put out by a range of groups, brackets, one of which including me, that in fact we're all saying that in fact maintaining patients with a restrictive fluid approach near zero balance leads to a decreased complications, hospital length of stay. That was the uniform guideline across all bodies. And we in fact showed exactly the opposite. In actual fact, there was no reduction in complications. There was absolutely no reduction in hospital length of stay. And in fact, we found markedly higher rates of acute kidney injury, wound infection, need for renal replacement therapy, even after what is relatively modest surgery. So that only came out 2 months ago. We've already had 15 citations. So I think we're going to actually lift the impact factor of New England Journal in the next 2-year period. And it's had great exposure, of course, because it is a really big deal. 10% of the population, of the Western population, have major surgery every year. In Australia, 1.5 million people have major surgery. This question is not just for abdominal surgery. It's really all types of major surgery. The mechanisms are all the same. And I've spent a bit of time enjoying the sunshine of Europe in the last few months promoting the findings of the study. And it's clearly, I'm told by many, many people, markedly changed their practice more or less overnight. So our ANZCA Clinical Trials Network are really trying to design, come up with what are the variations in practice, the important questions across all types of surgery and so on. We're aiming to design what we see as practical clinical trials. They are all multi-centre, they're all multinational, and they're seen as, we want to do large-scale trials so that we can do meaningful subgroup exploratory analyses and so on. Very pragmatic design, very diverse settings, trying to enrol as many eligible patients as possible into the trial. We allow local practices otherwise, but we collect those data and we analyse for it. And that was particularly important for the RELIEF trial when many people were criticising the trial because they said, oh no, in our practice we're so good at doing X that these results don't apply. So we analysed for X or no X, no difference. We did it over and over again with every single excuse people could come up with at that time. Now, just one thing I want to sort of briefly talk about, what's been one of my bugbears over years is when we look at clinical trials, and particularly in anaesthesia or the perioperative setting, is in fact everyone measures things a bit differently, whether it's post-operative myocardial infarction or post-operative renal failure or post-operative sepsis or surgical site infection, whatever it might be, they've got their own definitions. It literally makes it impossible to benchmark or compare studies and even more importantly to actually do useful systematic reviews and pooled analyses. So I've co-led this international group called the STEP project where it's standardising endpoints in perioperative medicine, use a range of really high-profile trialists from around the world. Where we've had meetings in a lovely place in Italy, and we've had a lot of teleconferences. We've put forward a design paper which was published in BMJ Open, and we've now published 3 of the major theme groups following the Delphi consensus process. They're coming up with these are the predefined, what we believe important endpoints when you're going to study any of these areas to do with perioperative medicine. And I think it's been a major advance, or will be a major advance, in anaesthesia and surgery. Now, just to finish off for a couple of minutes, I tear my hair out all the time trying to work out how can we make our research more practical, more useful, more informative for our consumers, which is mostly actually the anaesthetists themselves, perhaps other perioperative physicians, not so much the patients more directly. And there are exceptions, of course, to that. But overall, as we all know, less than 5% of hospitalised patients are enrolled in a clinical trial. And what are the impediments to that? And there are many of them. But some of them are things like the fact that we're all too busy and we haven't got the time to screen patients, enrol patients, consent patients. And I can tell you in elective surgery or emergency surgery, you've literally got 30 minutes. They come in the hospital, they get attacked by everybody, and they've got to be in the operating theatre in 30 minutes' time. It's not an easy environment to consent and randomise patients. Same time, clinicians have got their own point of view or preferences where they believe their approach or their drug or their dose is so much more right than someone else's. And that's prevalent. That variations in practice is everywhere. And of course, they often have their own preferences around it, including the patients. So trials are very expensive. There's slow recruitment. There's often questions around external validity. So I've in fact tried to deal with this issue with a new trial design, which I call the practice preference randomised consent design. And this paper has only come out a week ago in the American Heart Journal. And it really is the rationale and design paper of a big randomised trial where we are, in fact, going to do in cardiac surgery. Because in cardiac surgery at the moment, in different parts of the world, some centres or some countries give all their patients high-dose steroids as part of a sort of anti-inflammatory regimen to improve recovery after surgery. And other countries or other settings never use it. I can tell you in Australia, no one uses high-dose steroids in cardiac surgery. In the Netherlands, everyone uses high-dose steroids in cardiac surgery. And there's some countries where it's quite patchy. So this variation in practice, which is the sort of the enemy of high-quality healthcare, I think can be harnessed in a research design so that you include that local preference, the clinician or patient preference, and randomly assign in an unbalanced way 2-to-1 favouring the preferred practice, and using the Zelland's pre-randomised consent model, which has been justified, then in fact you only need to consent 1 in 3 patients. The others are automatically enrolled into the trial, and we have a partial assumed and deferred consent model that makes that ethical. We've had the study approved by our ethics committee here at the Alfred, and therefore it can run nationally through the national approval process. Equally, in the Netherlands, which has got very stringent rules around ethical and patient consent, it's been approved and it's now running. We pilot tested it here at the Alfred Hospital. In fact, ended up with a 98% consent rate for patients having cardiac surgery. So we can now enrol a broader spectrum of patients highly efficiently at much lower cost than a traditional 2-arm parallel trial. Now, at the moment, that's all theoretical, but I believe it is part of a solution of how we can make our trials have more impact and easier implementation in the future. Thanks very much.